Hi everyone, this is Kalil from Catalyst Learning Center. Do you know that behind every strong heartbeat and every deep breath, there is an unsung hero at work? And this is histidine. This remarkable amino acid fuels vital enzyme activities and helps build red blood cells that deliver oxygen and white blood cells that guard us against infectious agents. Without histidine, our body's defense and energy system would simply fail. So, by the end of this video, you'll be able to
First, describe the general structure of an amino acid. You'll know all about its four main parts: the alpha carbon, amino group, carboxyl group, and the R group, and understand how they are all connected and what they signify.
Second is to identify and classify amino acids based on their side chains. You'll be able to recognize how the R group makes each amino acid unique and how to group them as polar, nonpolar, acidic, or basic.
And lastly, you'll be able to explain how amino acid structure affects protein function. You'll understand how the shape, charge, and polarity of amino acids influence how proteins fold, how they work, and why they're so important in keeping your body running smoothly.
After watching, you won't just know amino acids. You'll be able to confidently talk about them and see how they bring proteins to action. Let's get started. Every amino acid has the same basic skeleton, kind of like a universal template, and it all centers around a single superstar. So at the heart of every amino acid, we have the alpha carbon. Think of it as a central hub in a busy airport. Everything connects to it. It has a tetrahedral geometry, which means it has a 3D shape with 4 different groups sticking out. Fun fact, this carbon is chiral. It has handedness in most amino acids except for glycine, which is the chill one without any handedness because the side chain is just a hydrogen. Next, we have the amino group, written as NH2. This is a basic functional group, meaning it can grab onto an extra proton when needed. In body's normal pH around 7.4, it usually grabs a proton and becomes NH3. You can think of it as a proton hugger of the group. On the other side, we have the carboxyl group, or the COOH. This one is acidic, meaning it tends to give away protons like a generous friend. At a physiological pH, it loses a proton and turns into COO. So, one side accepts proton, amino, and the other donates them carboxyl. They balance each other out. Now, for the star of the show, the R group, also called the side chain. This is what makes each amino acid unique. Like different outfits in the same mannequin, the R group determines whether an amino acid is polar, nonpolar, acidic, basic, or anything in between. It also affects how the amino acid interacts in proteins, including how it folds and what function it performs. Finally, we have a single hydrogen atom bonded with the alpha carbon. It might seem simple, but it's crucial to complete the tetrahedral shape. Without it, the structure wouldn't be stable. So, what actually happens to amino acids at physiological pH around 7.4, also known as the pH inside your body? They exist in a special form called zwitterion. First, the amino group grabs an extra proton and becomes NH3. Meanwhile, the carboxyl group loses a proton and becomes COO, like dropping off an extra baggage. As a result, the molecule has both positive and negative charge at the same time, kind of like a walking battery. But here's a twist. The net charge is usually zero, because of the positive and negative charges cancel out each other. This zwitterion forms a super important of how amino acids dissolve, interact, and move around in your body. Next up, how do we group amino acids? We can classify them in 4 big categories. First is the nonpolar, or the hydrophobic. These are the water haters. Their R groups are nonpolar. They avoid waters like to hide inside proteins. Think of them as the shy ones at the party. Next, polar, the uncharged. These side chains can form hydrogen bonds and love hanging out with water. They're friendly, but don't carry a charge. Next is we have the acidic, the negatively charged R groups. These guys have the R groups that lose protons, giving them a negative charge at a physiological pH. They're the proton donors, the super generators. And lastly, we have the basic, the positively charged R groups. These side chains grab extra protons and carry a positive charge. They're the proton collectors, always looking to pick up a spare. So today, we saw how amino acids act as zwitterions inside your body, and how we sort them into groups based on their side chains. Next time you think of proteins, you just remember all this starts with these tiny, versatile building blocks. And that's all for the Catalyst Learning Center. Thank you.